So today I am going to be talking to you about some of the work I have been doing as part of my PhD, which is a collaboration of fusion antibodies here in Belfast and is kindly funded by the Medical Research Council. So just to get started, quick overview, I'm going to run you through some important background for what I'm talking about today, the aims and objectives of the work shown here, some of the results, a few of the conclusions we've drawn, and some of our ideas for future work. So to start, I know people don't always like saying these pathways, but death receptor 5 or DR5 is our first target of interest in this dual targeting approach. Just for those of you who wouldn't maybe be familiar, DR5 is the starting point of the extrinsic apoptotic pathway. So it's here. Apoptotic signaling is triggered by the oligomerization of the receptors induced by the binding of its exogenous ligand trail. We get the formation of what is known as the DISC, or the death-inducing signaling complex, which is comprised of various adapter molecules like FAD and these death domains internally on the receptors, which recruits pro-caspase 8, which undergoes cleavage to its active form and then subsequently activates caspase 3,7 and triggers apoptosis. Our key inhibitor of this pathway that I want you to keep in mind for this talk is FLIP. And why we're interested in this pathway is DR5 expression correlates with poor prognosis and survival in several cancers, such as pancreatic and colorectal. And for example, in pancreatic, we see a 5-fold increase in DR5 surface expression in cancerous versus normal tissue. So this vast upregulation makes it an attractive therapeutic target. And a range of monoclonal antibodies have entered clinical trial, 6 in total, but none of them have managed to progress beyond phase 2 trials due to lack of efficacy. So now these so-called second generation DR5 agonists are exploring alternative antibody formats to try to overcome these efficacy issues. I suppose the efficacy issue can be boiled down to the fact that a traditional antibody is bivalent and the active signaling unit for DR5 is the oligomerized receptor. So in essence, it's very hard for something that can only bind 2 sites to bring more than 2 sites together. So it's often insufficient to induce effective apoptotic signaling alone. So previous work in the Scott lab has shown that these antibody conjugated nanoparticles are much more capable of inducing apoptosis in a range of human cancer cell lines. This clustering effect from the antibodies on the surface of the nanoparticles allows for efficient receptor clustering, oligomerization, and subsequent signaling. So we've shown this in a range of cancer cell lines in Scott group. And I suppose the aims and objectives of the work I'm showing you today is previously we'd only used one human DR5 targeted agonist, and we wanted to see if this platform is transferable using different DR5 agonists in murine cell lines. Can we manipulate and enhance this targeting effect using chemotherapeutics? And then the advantage of having our murine lines in syngeneic models is we really want to have the opportunity to unpick the mechanism of action of these dual targeting particles. So to start, we did this in a range of cancer cell lines, but for the purposes of today, I'm only going to talk you through results in 2, MC38s, which are colorectal line, and PAN02s, which are pancreatic. And first of all, we demonstrated via flow cytometry that both of these cell lines express very high levels of DR5 in their cell surface, and they also express a key range of pro and anti-apoptotic proteins, key proteins in the signaling pathway. Again, our key ones that I want you to bear in mind is this presence of FLIP, which is an anti-apoptotic protein, and then we also have our procaspase, which is important for actually executing apoptosis, and FAD are important adapter proteins. BAX and BCL2 aren't hugely relevant to our story today, but it was important that we established that they were present. So then moving on to our actual DR5 agonist, the antibody we're using is clone MD51, which was a kind gift from Dr. Hideo Yagita in Jamendo University in Tokyo. We used single emulsion evaporation method to prepare our particles. In our organic phase, we used PLGA and a PLJ PEG, with NHS functional groups conjugated. I won't labour the method, as I'm sure everybody is relatively familiar with this. And then when we had our PLJ-PEG NHS particles, we were able to add our antibody to our particle suspension, and with simple stirring at room temperature over 2 hours, the NHS and amine groups on the particle allowed amine coupling reactions to occur, and we were left with conjugated particles that we washed via centrifugation. See, we've quite favourable characteristics. There's very little difference in size and PDI for these particles, and we're getting a conjugation efficiency of about 30 to 35%, which we considered reasonable. So our next step was to see, can these antibody decorated particles induce apoptosis in our cell lines? So we have our, once again, we have our MC38s and our PAN02s. We use cell titer glow at 24 and 72 hours. Here our line of interest is this red line. These are our antibody decorated particles. And then we also have our free antibody and our blank nanoparticles, neither of whom are able to induce any meaningful reduction in cell viability. Our nanoparticles in the MC38s were shown to maybe reduce viability between 25 and 30% at 24 and 72, and much more variable, probably only about 10% in the PAN02s. We also looked at some key apoptotic or cell death protein via Western blot after 24 hours. So PARP cleavage is a classical marker of apoptosis, which we can see here in the MC38s with our antibody-coated particles, but not with our free antibody or a blank particle. We're also seeing the cleavage of caspase 3, indicating its irreversible activation and drive towards apoptosis. And we're seeing possibly some inhibition of FLIP, which we will move on to a little bit later, why this is important. We're not really seeing any of it in the PAN02s, which kind of lines up with the cell titer glow data that they're a bit more resistant. So we've established we have a little cell death, but not a lot. So our next question was, how can we seek to improve this? So it's not one, but dual targeting. So previously we have shown that camptothecin is capable of reducing viability in these murine cancer cells. Camptothecin is a topoisomerase 1 inhibitor, prevents DNA religation during replication, which eventually causes the induction of double-stranded breaks and apoptosis. It's a common chemotherapeutic drug that we are familiar with working with in our lab. So camptothecin is quite hydrophobic, so we were able to successfully encapsulate it into our nanoparticles and also successfully subsequently conjugate our antibody onto their surface. Do this by adding CPT to our organic phase in our particle preparation. And we can see that we have particles of very uniform size, a bit of variation in PDI, but importantly, the encapsulation or entrapment efficiency of CPT remains very similar for our nude and our CPT MD51 particles, and our conjugation efficiency shows little variation in these particles as well. So our next step was to take this forward and to put it on our cells. So here our important line is our purple. This is our CPT loaded MD51 coded nanoparticles. Our blue is our CPT particles with no conjugating ligands on the surface. Red, as before, is our antibody coated particles with no drug entrapped, and black is our blanks. And as you can see here in the MC38s, we see this increased cell death versus the antibody coated particles or the CPT nude particles. See this increased, maybe an extra 20% cell death, and at 72 hours we still see this increased sensitivity at low concentrations. Once again, like we'd previously seen, the PAN02s do not seem to show any appreciable sensitivity to this. They stay very much the same as the CPT particles, nothing really significant. So our next focus is now trying to understand, why are the MC38 cells sensitized by this combination, and why aren't the PAN02s? So to bring you back to our pathway here, as I mentioned before, the key inhibitor that we're looking at here is FLIP, and we have shown that camptothecin inhibits FLIP in our MC38 cells, but not our PAN02 cells. And so what we think is driving this sensitivity in the MC38s is when you knock out FLIP, you're effectively cutting the brakes on this apoptotic pathway. So in essence, you've cut the brakes, and with our DR5 binding particle, we're also pressing the accelerator. So we're getting a vastly amplified apoptotic effect compared to either particle alone. For whatever reason, the PAN02s seem very resistant to this CPT inhibition. However, it's really worth noting that, relatively speaking, the PAN02s express a lot more flip. So it may just be that, as camptothecin is not a specific flip inhibitor, that its mechanism of action may just not be specific enough to inhibit flip in these cells. So we have been exploring more targeted novel flip inhibitors in our lab to see if we can overcome that issue using those. And then, so what we have shown here is that our CPT-loaded MD5 encoded particles sensitize our MC38 cells and help overcome apoptotic resistance. We want to unpick and explore the differences we've seen between these sensitive MC38 cells and the relatively insensitive PAN02 cells. And then the real advantage of these cell lines as they're murine is we want to try take these forward to an in vivo study, and we want to look at differences in the effects they may exert over the tumor microenvironment, as previously we've not had the opportunity to use syngeneic models. So we wish to look forward and see what we can do there. So just to acknowledge everybody who's helped and facilitated this work. And yeah, I'm happy to take all the questions there.
Thank you, Anna. Okay, so open for questions. Tom, you have a question?
Yeah, please. Yeah, thanks, Anna. Really interesting talk. I was wondering if, I don't know if you've looked at this yet, but it seems a really interesting question to me in terms of how the particle properties influence the presentation of the antibody to the cells. So do you think like smaller particles would help lead to more binding, or the peg chain length is your spacer? Would that influence how you present the DR5 antibody to the cells?
It's not something we have looked at, but it's very possible if we have other people in the lab have looked at these modified antibodies to give better presentation, like Fab fragments and stuff. So it is very possible that our particles are, whatever way the antibodies are binding, because it's not a site specific, that they are being occluded, potentially increasing the PEG linker to push them further out from the surface would allow them to orientate better. It's not something we've looked at, but no, it's an interesting angle to take from it.
I guess with all nanomedicines, there's so many different variables, aren't there, to change, that you've got to fix it somewhere, haven't you? I was wondering also, when you've got the camptothecin in the particles, did you actually see, or were you able to look at the accumulation? So was the presence of the antibodies helping to improve the delivery of the drug into the cells? I might have missed that.
In terms of the particle uptake by the...
Yeah.
I haven't looked at it, but previous work in the lab has shown that those active targeting effects, certainly in our human cells, has improved the endocytosis, because DR5 will endocytose when it is fine. It has improved it, but not specifically in the murine cells, but it could be a part in why the PAN02s are more resistant if they're not doing that.
Okay.
I agree.
Cool. Well, thank you. Really interesting talk, Anna.
So a load of questions coming in here in the Q&A. Are you able to measure the degree of clustering of DR5, or is there a minimum threshold that needs to be reached, do you think, Anna?
Ooh, there probably is, in that what we have found with these particles is there's a degree, it's like an amount of polymer, I suppose 0.6 of these cells is what we've needed to see that induction. So there obviously is a degree of oligomerization that you need to get that effective signaling, but we've only ever measured it as a proxy of like polymer and particle concentration. Your increased in conjugation of nanoparticle, like antibody on the nanoparticle, hasn't been shown to make as much of a difference, but that's probably coming back to the fact that if you're trying to put too much antibody on, you're going to get more of it misorientated, so there's actually less effective binding zones. So there definitely is a threshold, but we've never looked at it in the sense of the receptor, more just on a proxy via polymer concentration.
So that comes on to a next question. There are actually two questions here. I'll try and combine them. So how do you confirm that the orientation is correct? And did you look at the stability of the particles?
Stability.
You try and merge those into one so we can move on.
So we have looked at our general stability studies via making the batch particles, measuring their characteristics over a period of time, side DLS, conjugation, and our entrapment efficiencies. With the orientation, there's no real way of monitoring what's happening in the vials, but by virtue of looking at where the sequence and stuff, where those side chains are, you can be relatively confident of a certain, like an equivalent or necessary percentage orientating correctly. But I don't know how necessarily you would confirm how many antibodies you're actually getting orientated exactly right, and what sort of microscopy you would need to be really diving into how many of them are being presented properly. But
Okay. Well, we can do site-specific modification things to orientate and enhance that. So Anna, thank you very much